All right, so let's begin our review of really everything that we covered this year, um, with the exception of the things that aren't going to be on this 2020 AP Biology exam. And of course, this is a little different than usual. Each year we talk about how there's so much we've covered, and some of it will show up on the test, and some of it is bound to not show up on the test. Well, this year, more than ever, that's true, right? That we covered a lot this year, and it's hard to say exactly what will show up on these couple of questions that we're going to have. But we'll still review over everything that I think could matter and could show up, and we'll kind of highlight things that I think might be most important to keep in mind, and we'll just do the absolute best that we can. So, first thing is formatting. So, just a few comments. I always kind of like to start our end of the year review with this information, which also is going to be different for this year in 2020. But in terms of our format, right? So, you are, and we've talked about all this stuff too, I think, before, but you're going to take an online test. It's going to be Monday, May 18th, starting at 2 p.m. You can complete this with any device at all, computer, iPad, phone, even. And you'll have two options, is what they're saying, which is that you can type your responses and just submit, or you can write your responses out, take a picture and submit that or upload that, which I assume is the way that it would work if you were trying to work from a phone. The test is going to consist of two free response questions, one long free response and one short free response. The long free response question, they said, will be of the interpreting and evaluating experimental results type, which is not a surprise to me at all. I think the major focus of this year's test is going to be looking at experimental results. And the short free response is going to be a conceptual analysis question. Now, they also released just recently the weighting, so it'll be a 65% of the score will come from the long free response, and 35% of your final score will come from the short free response. It's still hard to exactly know what that's going to mean. As we've seen with practice questions, long free response are typically 10-point questions if we're talking about a raw score, and the short free response questions, at least of this type, are typically 4-point questions. So how that's going to weight out, I don't know exactly how they're going to do that, but they will, and we'll see how that turns out. Now, technically, even though they've been saying it's a 45-minute test, you're technically going to have 50 minutes that you'll be working. The long free response question, you're going to have 25 minutes to answer, and then you're going to have a 5-minute window of upload time or submission time, and then you're going to take 15 minutes for the short free response, also followed by a 5-minute upload time. So active working is about 40 minutes. Then you'll have this 10 minutes of uploading time as well. And as we've talked about, the exam this year does not include College Board Units 7 and 8, which are deeper looks at evolution and ecology. So those topics could show up kind of at a surface level, but a lot of the depth that unfortunately we had covered in the end of the third quarter, start of the fourth quarter, will not really show up on this year's exam, or shouldn't. Now, I thought you might wonder about this as well. What is interpreting and evaluating experimental results, and what is conceptual analysis? And so I pulled this up from the College Board AP Biology website. Both of these question types, of course, are going to start with an authentic scenario. So, they're going to describe some sort of biological setup. Obviously, for the longer free response, this is going to be accompanied by data because it will be a discussion of some experiment. Then with that, they expect you to be able to do a couple things, right? Describe and explain concepts. Well, that's everything, right? But it does say in applied context. So what that means is, you need to be able to explain kind of what's going on in that question. So even if the question is an experiment, there's a good chance that you're going to have to understand the context of that experiment, whether it be genetics or cell signaling or cell division or metabolism or whatever that might be. You'll have to justify experimental design procedures. So why was the experiment designed like it is? You'll probably have to analyze some data and give some sort of explanation of what you see in the data. And then you have to make and justify predictions. And again, remember, you're going to see this justify term a lot, which just basically means support your answer. And so if you're justifying with data, make sure you state specific data. If you're justifying with, I don't know what else, you'll just make sure that you support your answer that you say. And so if you are looking at old exams or things like that, what they're saying is that this will be like a traditional exam free response question number 1. What I've found in looking at old tests is that just more recently they've sort of streamlined things where question number 1 is the interpreting and evaluating experimental results question, and question number 2 is doing the same thing but with graphing. It wasn't always that way, and so when you're looking at old tests, look at numbers 1 and number 2, which might both be good representations of this type of question. But as I've said previously, I don't know that we're going to be expected to graph this year because it's kind of hard to type a graph. But we're going to do 1 or 2 practice ones still with graphing involved, because I think if you can make a graph, you can definitely interpret a graph, so it's still really good practice. Now, the other side of this, I'm taking way too much time here, but conceptual analysis might not necessarily be an experiment. It could be, but what they talk about is a biological phenomenon with a disruption, meaning some sort of thing has happened, and they're going to tell you that. Might be based in an experiment, or it just might not. It might be something that's observed in nature or whatever. And they want you to, again, be able to describe and explain that in context. So whatever the topic may be, they still want you to be able to make predictions, right? And they want you to be able to support and justify a claim. So notice that these are really similar things when you really break them down in what you're expected to do. And again, they've said that this is going to be like traditional exam FRQ number 4. What that definitely tells us is that's the traditional kind of 4-point kind of question, which generally has 3 parts to it, right? The 4-point questions are usually parts A, B, and C. Again, what I've found looking at previous tests, I think questions 3, 4, and 5 could all be of a type that we would expect. Again, in just the past year or 2, they've really streamlined this where number 4 is always the same kind of thing. In the years previous to that, questions 3 and 5 are also really similar. So again, if you were looking at some old tests online or something to look for some practice problems, I think questions 3, 4, and 5 could all be potentially helpful to you. Now, I always like to offer my own tips as well that I think might help you on your way, and that goes well with formatting. So here's 2002 Mr. Burton. Haven't changed a whole lot wider now. A lot more hair on my face, I guess, too. But I knew what I was doing, right? I got a 5. But notice I put here, I barely knew what the internet was. That's a little bit of a joke. I mean, I guess Facebook had just been created probably in 2002. But we certainly were not doing any of our schoolwork online. We might have been doing a little bit of research or something here and there, but nothing like today. And there were certainly not iPads or anything either. So if all of this had happened when Mr. Burton was a junior in high school, we certainly would have just not had school. The year would have been canceled, and AP exams most certainly would have been canceled as well. But so, some things that I would suggest, and these are not, things that you have to necessarily do, but things to think about. First thing is, you got to know your notes. There's this temptation when a test is open book, which this technically is, to think that you don't have to study quite as much. But you may have learned in school already that a lot of times open book tests are harder, right? And that could be the case with this, right? They might make this a difficult couple questions because they know that people can access their resources. And so in some ways, that might mean that they try to push us more than they ever have before. I don't know that that'll be true because of the situation that we're in, but it could be. So prepare. right? Go through as much as you can stand all of our notes that we've done, and use as many resources as you can to do some practicing and things like that. Second thing, though, is be prepared to cheat, right? Quote unquote. Again, this is open book. They've talked about doing everything possible to ensure that students are doing their own work. So please don't try to come up with some complex way that you're going to work with other people from class. If they are to be believed, they're going to have ways to sort of detect that. And if they, I would say, have any inkling that you have done something like that, they're just going to avoid your test altogether. So that's not what you want to happen. But what you can do is surround yourself with resources, right? So I would suggest that as you look through the old notes that you have, make some study sheets or even posters to put around you during the test. Maybe it's a cork board right in front of you with a bunch of stuff on it, or maybe it's posters that you tape on the wall, so that you can quickly access some of the basic information or some of that information that you don't feel like you'll remember. Use Post-its to separate stuff. I realize that most of your notes are in your iPad, but if you've got your textbook or other paper resources, or you make enough study sheets, use little tabs and stuff so it makes it more quick, to move from section to section or whatever it might be. And then I'm kind of old school, but I think you're going to want some scrap paper, right? As you read the question, particularly in an online setting like that, it might really help you to jot down things on some scrap paper, thoughts you're having, kind of outline it, and then transfer that to your actual question that you're gonna, or your actual response to the question. Another thing that I'm thinking, and I know this might just be something that you can't, get around, but if it's possible to use a secondary device, I would do that, right? For me, I'm much more comfortable like typing on a computer. So if I was going to take this, I would try to have a computer that I take the actual test on, and then I would have my iPad there to access for my notes and everything else. Obviously, I know that's an ideal situation if you have more than one device, and that might not be something that you can do, but definitely something to think about. And if you're in a situation where you can't get a second device and you really know you would want that, reach out to me. I'm sure that we might be able to work with school to try to get a loaner device or something like that in your hands so that you're not trying to bounce back and forth between the test and your notes or something on your iPad. And then the last couple ones are just a little sillier, but also still true. Remember, ATFQ, right? Answer the freaking question. Um, uh, you know, Again, now more than ever, there's only two questions, and so you don't want to write a whole bunch and just not really be answering what they're asking you for. So pay attention to those bold words in their question that really kind of direct you to what they want from you. Keep in mind that the short free response is probably not going to require, or won't require as much detail as the long free response, because each piece of the question is only going to be worth like 1 point. So, hit it and quit it. Get right to the point, make your point, and move on. The other thing that I haven't really said this year, but I put in here, MSU, right? Make stuff up if you have to, right? The worst thing you can do here, particularly with just two questions, is leave anything blank or unanswered. So just make something up if you have to, right? And like I put here, you might really surprise yourself and what accuracies you drop in there to get at least part of the credit. You know more than you think you do. I promise. I promise you do. Every one of you guys have been incredibly devoted this year and you've worked your butts off. It's in there, right? The information is in your brain somewhere in most instances. And so even though it might not float to the top, if something strikes you, just write it down. Um, again, you might be shocked at what actually comes back from that. And then the last one is have pride in yourself. Again, you guys have worked your butts off this year. You are capable of a lot. And regardless of how well you personally have scored on a particular unit test in our class, right, you still have every reason to be confident. Remember that you are scaled against everyone else. That's really hard when you're talking about a class of less than 20 people, right? But when you look at a huge group, realize that our 19 students are going to be toward the top of that group, right? So don't give up, right? I put this is something that one of my college professors used to talk to us about, those of us who were getting ready to go to medical school, because that's so challenging. He said, don't give them the pleasure of you giving up. That's what he used to say. And at first I was like, what? But it's a good point, right? If you give up, you've given in, right? And I'm not saying that the AP people are out there like trying to make you fail. That's not their goal. But it sort of is, right? That's why they make their test hard. And so don't give up. If you give up, they win, right? So be proud of yourself, have confidence in yourself, and that can go a long way. All right, so we're gonna run through some topics, and I'm feeling like this first one, this quarter one review, is probably gonna take the longest time here, by the way, also. But so the first thing, I think this is gonna be incredibly important, right? Basic experimentation. It is the long free response question, basically, with some further details from some content that we've learned. But it's definitely going to be about an experiment. So you got to know about those experiments. First thing, hypothesis. Right, and I kind of talked about this, I think, with one of the free response kind of practice questions we had. But when they ask you to make a hypothesis, sometimes the question's going to be worded a little funny. But basically what they're always asking you is why, right? So make a hypothesis for blah, blah, blah. What they're saying is, why does blah, blah, blah happen, right? And so you're going to provide that why, that background, that because with your hypothesis. Second thing, you may have to highlight, or it might just even ask you, what are the independent and dependent variables? So remember, right, independent variables, or sometimes called manipulated variables, are what you are changing in each trial, right, or what the scientist is changing from group to group or from trial to trial. You're going to find this in your setup of your experiment, right? How are the trials set up? Therein lies that difference, that independent variable, versus dependent variables, which is what changes as a result. So this is what you're going to see in the results, right? What do we expect to be different because of our independent variable? Now, next thing, control group, right? You might have to point out the control group. They might ask you, why are you using this particular group? And what's most important is being able to explain a control group. Right, so a control group is there to show that the independent variable is responsible for whatever changes you are observing. Right, now one thing I've done you guys a little bit of a disservice is we didn't really talk about positive versus negative control. Right, so we've got positive control, we've got negative control, and I'm confident you could figure this out if it came into play. But positive control basically confirms an expectation. So I'm at the moment kind of having a brain fart, and I can't think of a great example of this. But a positive control is where you do a trial, and you treat something a certain way, and you expect a certain something to happen. And then your experimental trials are where you remove that stimulus, right? Now, that's a little less common than what we typically work with, which is a negative control, right? A negative control is where more so we expect nothing, right? So, for instance, let's say you've got two trials. In one, you're going to use this insecticide that will hopefully get rid of bugs on your crops. In the second trial, you're just going to use water, right? In that water trial, you expect nothing to happen. You kind of have a no treatment group. And that's a negative control. And again, that's what we typically roll with. But sometimes it makes more sense in an experiment to use what's called a positive control as well. Now, also data. I would say that more often than not, you're just expected to analyze data. But sometimes you're asked to devise an experiment, right? And so you also want to think about that. That's a possibility. How would you collect data? Most important thing with collecting data is making something quantifiable, right? Make sure that you can count, you can measure that data that you're taking. Data that you can't measure is sometimes helpful, but if an experiment has no quantifiable data, it's probably not a very good experiment. There could definitely be supportive data, that's qualitative, right, not quantitative, but quantitative data is definitely better. Again, probably not going to have to present any data because that would always be graphing, but definitely being able to analyze data is important. And then don't forget that more data is better. We talked about standard error when we looked at statistics. And remember what we said is that standard error decreases with more data. So in other words, more data, less error. And, you got a better experiment that way. And then we have conclusions and predictions. We can pretty much almost guarantee, I would say, that there will be some piece of one of our questions that talks about some predicting or some conclusion drawing. Typically, they're going to ask you to justify this, right? So if you're justifying again, that just means support your answer. And if they say justify with the data, which is often what they do, then make sure that you cite specific data. Before we leave this slide, this is a good opportunity also to talk about something that has become more and more common in the last couple of years, and so I need to start teaching it better. And that is to put error bars on a graph. So basically, what that means, right, is you end up with your bar, and then they draw something like this on it, right? Well, what's going on here, and obviously this is a very generic graph, but let's say that, this is a bar represent 5 of something. What this would be is let's say that you are told that you have a standard error of 1. All right, well then what you would do is you would say, okay, I've got my data point at 5, and I'd put my error bar at 6 and 4 to show that, with error considered, this data really falls into this range. Also, sometimes what they might say is instead of saying standard error is 1, they might say 2 times the standard error. Right? In this case, if we're working with the same problem, that would just be 2. And so then you just make sure that when you put your error bar on here, the error bar has a distance of 2, right, around that data point. So could be something you see in the graphs that show up in a question. All right, graphing and rates, our second topic. Now, traditionally, we would talk a lot about how to make a good graph, right? That's not as important, I don't think, this year. But don't forget dry mix still. Certainly, they could have a graph and ask you about an independent variable, and so an easy way to identify that would just be to look at the graph, right? So remember that the dependent variable typically is on the y-axis, while the independent variable is typically on the x-axis, or dry mix. Also with graphs, again, this is more helpful when you're making the graph yourself, but if the data is connected, if it's over a period of time, if it involves rates at all, then you're always using line graphs. If you're just counting your data, documenting what happens in sort of different scenarios, then you typically would go with a bar graph or a histogram. One thing that I wouldn't be shocked to see show up on our test, and so because of this, I've been testing you on this all year long, is rate, right? Rise over run, or the change in y over change in x. So technically, this is the way we would write this formula, right? Or y2 minus y1 over x2 minus x1. And again, they're typically going to say, okay, what's the rate of change between 0 and 20 minutes? Well, time's always on your x-axis, right? So we would say, okay, from 0 minutes to 20 minutes, and then whatever we're doing up here at the top, we would also plug in and then solve for that. So rate, not guaranteed, but pretty decent shot it could show up. Topic 3, the chi-square test. Now, realistically, I'm not so certain that they'll bring in other statistical analyses. I know that we covered several different things like standard deviation and different things at the start of the year, and I don't think it would be a bad idea to check that out. So if you go back, kind of look at your notes, just have a rough idea of what standard deviation is and standard error and things like that. But I think the most likely thing that could show up, because it would have the tendency to trip up more people, is a chi-square analysis or a chi-square test. So, here's our formula for chi-square. I also don't really know how they'll handle the formula sheet. I kind of assume the formula sheet will be provided within the online test, but it might kind of be harder to access. So one thing you might want to do, I've got that formula sheet posted on the Schoology site, just like on the kind of home page. You might want to print one of those off for yourself, or if nothing else, just write down some of the things from it that we've used this year so that you do have access to those for sure. But the chi-square formula, remember, what we're going to do is we're going to sum up sort of an evaluation of each of our data points, observed versus what we would expect to happen. And remember that the chi-square test is really asking how good was our hypothesis. And we talked extensively at the start of the year about a coin flip, right? We expect a coin flip to always come out 50-50, but the question is, will it really come out 50-50? And so we always start with a null hypothesis. I think this is an important term to know. I could see them asking you to give them a null hypothesis and then not even going through the entire mathematical process of a chi-square test, but just giving a null hypothesis. And remember what this means, right? Null meaning nothing. So the null hypothesis is sort of like nothing out of the ordinary, right? So again, if we flip a coin 200 times, our null hypothesis would be 100 heads and 100 tails, because we know that there's two sides to the coin. Classes is the number of possibilities. So again, with a coin flip, we'd have heads or tails, so it'd be two classes. And then degrees of freedom is a calculation that we would use statistically, and that's the number of classes minus 1. So we've got to be able to use this formula and use the probability table of statistical values that we are given. Again, remember that the O represents the observed value, so this is like actual data, while the E represents expected value, or sort of like our hypothetical expectations that we would have for this. Then here is our statistical table that we mentioned there a minute ago. We always use this 0.05 row as it is drawn on the formula sheet. Remember, what we do is we compare our chi-square value to, whatever is in this chart for our particular degrees of freedom. So again, with that coin flip, we had talked about 2 degrees of freedom. So whatever our chi-square value came out as, we would compare that to this value of 5.99. And then if you're asked to kind of justify your prediction or justify your analysis, we've got to know what these number comparisons mean. So if the number you come up with, right, your chi-square value is greater than the number in the table, then you would reject your null hypothesis. Think of this as being too big, right, your chi-square number. And what this means is that you are not close to expectations. So something is up, right? I mean, something is out of the ordinary in this phenomenon or this scenario, because it's not coming out as you expected from a statistical standpoint. Now, the other option here is that your chi-square value that you came up with is less than what's in the table, in which case you would accept the null hypothesis. And this means that your data is close enough to expectations, and any minor differences are simply due to chance. So let's do a practice chi-square problem here. Put a little blank slate up, and I'm gonna kind of make this up as I go. So this could turn out poorly, but hopefully it'll work out just okay. So let's say that we're looking at peppered moths. Remember those guys from evolution? Basically, we can have let's see what a moth looks like here. We could have the light colored ones, and then we also had. I wonder if I draw in black, will it show up at all? Oh yeah, cool. Okay, so and then we have those dark colored ones, right? That just blend in potentially with their surroundings. And what we're told in the question is to assume that the light color is dominant while this dark color is recessive in these moths. And we're told that this is basically standard dominance, right? So this is Mendelian, where we would expect a true dominant and a true recessive form to show up. So the first thing they're going to say is, what would be your null hypothesis if we cross two heterozygotes? So notice how this is a good chi-square problem that I'm just making up here. So of course I think it's good. That deals with something realistic. Something in genetics is probably a good place to throw this at you. So a null hypothesis, if we cross two heterozygotes, realize what this is, right? This is just the Punnett square. So let's say two heterozygotes. Let's use M for moths, right? Heterozygous means one dominant allele, one recessive. So we cross, these two heterozygous moths, and we expect to get something like this, right? Or 25% homozygous dominant, right? 50% heterozygous, 25% homozygous recessive. So realize again, this is a way to establish a null hypothesis. This is my expected values based on just what I know about genetic predictability. Now, then you're told that in a laboratory setting, they actually cross two heterozygous, lightly colored peppered moths. And what they get is 8 out of 10 are light. And then they run some tests to break that down even more for us, right? They break that down and they can tell us that 2 moths were homozygous dominant and 6 moths were heterozygous, whereas the other 2 moths, right, the dark ones, are homozygous recessive. So check out what I've been given, right? What I've been given are some observed values. And we can also turn these into percentages, right? 20% homozygous dominant, 60% heterozygous, 20% homozygous recessive. Right now, what I suggest is use the whole numbers in this case for these percentages. So let's do a chi-square test. Right? Remember, we're going to sum up the observed minus expected squared divided by the expected for each of our classes. So first we'll look at homozygous dominant here, and so that's going to be an observed of 20 minus 25 squared all over 25. Right, then we're going to add to that our heterozygous class, or possibility, so that's going to be 60 minus 50 squared all over 50. And then we're going to do our homozygous recessive class. So again, 20 minus 25 squared over 25. All right, and so if we add this up, let's see here, that's going to be 5 squared. Okay, so this is nice. So this would be 1. Right, and 25 over 25, and then I would get 10 squared, 100 over 50, so plus 2, plus the same thing again. Okay, good. So this means my chi-square value is going to be 4. All right, so chi-square equals 4. Now, if we look back at our table, well, first, okay, so degrees of freedom. Remember, there's 3 possibilities here, so we'd say the number of classes is 3 minus 1, or 2 degrees of freedom. So for 2 degrees of freedom on our table, the value we had a minute ago is 5.99. 4 is less than 5.99, so we can accept our null hypothesis. All right, so even though this observed data here is not exactly like our expected data, it's close enough that we can accept statistically that this data makes sense. All right, so hopefully that's a nice recap of how we might use the chi-square analysis in our biological studies. All right, topic 4, enzymes. Enzymes are going to be such an important topic in biology. Again, we can't guarantee they're going to show up on this AP exam, but please try to remember some things about enzymes as you move on, because if you take a science class again, you're going to see them, right? And remember that with enzymes, like with most proteins, it's all about shape. And also put orientation here, so the ability to kind of come together, right? But remember that an enzyme, you want to know what it is, and you want to know what sort of situational things can affect enzyme activity. So remember, enzymes increase the rate of a reaction, so they speed up a reaction by decreasing the activation energy. And we see this great, graph here is a fantastic one to be familiar with. Might even be worth writing on a poster or a study sheet that you have in front of you. Remember, a reaction is going to start and finish at a certain energy level here. And under normal conditions, you've got this black curve. And under conditions with an enzyme, you've got this red curve. And the hill is smaller, right? It requires a lower activation energy to accomplish the reaction. And so remember, the way that it does this, right? The enzyme has an active site, and the active site has to be the correct shape to bind with a substrate. And once it does that, it will physically improve the conditions for a reaction. So it might stretch that substrate, it might bring multiple substrates together. Just kind of depends on what kind of reaction we're talking about. Now, we also have these things that we have to know about which would alter enzymatic rate. All right, remember that as temperature increases, that will typically increase enzyme activity or increase enzymatic rate, because it increases molecular movement, right? Molecules are more likely to slam into each other, which means they're more likely to interact with the enzyme, and they're more likely to interact with the other substrates of the reaction. Don't forget about this thing at the bottom, though, that we'll come back to in a moment with that. pH. Remember, pH, as we move toward optimum for that particular enzyme, that will increase the rate. Remember, every enzyme has an optimal pH. That might be high and that might be low. So for pH, we can't just say, well, raising the pH is a good thing or lowering the pH is a good thing. We have to look at that particular enzyme. And if the pH gets adjusted to closer to that enzyme's optimum, then we would see an increase in enzymatic rate. Substrate concentration. Remember, the more substrate we have, the more likely we're going to do a reaction. So it'll increase the rate. But remember that this is also limited, right? Remember that we can increase substrate concentration, but eventually we're going to get to a point where all the enzymes are filled. And so we'll see this thing level off, right? We'll see this graph level off because all the enzymes are working. Doesn't matter if we keep adding more stuff to work on. And then enzyme concentration. Again, if we increase the number of enzymes, that will also increase the rate. But this one also has sort of a limitation to it, and that is that eventually we run out of stuff to work on. So for this one, you're going to see your graph going, and then you're going to see your graph just go down, right? You're going to see it drop because the enzymes that we've added have worked their way through all the substrate, and there's nothing else for them to work on. Now, I mentioned there a second ago, don't forget about this thing at the bottom, and that's denaturing. Also, don't forget that they don't like you to just use that word, right? So what we can say is if we get temperature too high, if we get too far away from optimum pH, then we might denature. And remember what that means. Ultimately, it means that we change the shape of the enzyme, and therefore it loses its function. All right, but also remember that this protein enzyme changes its shape because the bonds break, right? The bonds that were holding that protein together kind of fail, the shape changes, and then the enzyme will no longer work. All right, topic 5. Do we need to know other chemistry? Man, that's a good question. I don't know. It's I guess possible that some other chemistry, kind of biochemistry, can show up. Um, I would say it's not the most likely thing in the world, but there's also a little bit of chemistry that it really underlies metabolism, right? And metabolism, I'd say, does have a pretty good shot of showing up. So, um, just remember some things, right? Organic compounds are mostly what we focus on in biology, and organic compounds are made of carbon. In fact, living matter, don't forget schnapps, right, is primarily 6 elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. Those things come together to form the major macromolecules, or the major biomolecules, which are carbs, lipids, proteins, and nucleic acids. And you just kind of have to know that. That should hopefully just be in the back of your head somewhere. Different types of bonding. I would be really shocked if this would show up on the test. But just in case, right, a covalent bond shares electrons. An ionic bond is an actual transfer of electrons. And a hydrogen bond is just more of a weak attraction than anything. Polar, nonpolar, I could see this kind of being just embedded in a question, and it might just help you if you understand what it's talking about. Remember, polar compounds dissolve in water. They almost have a partial charge to them, while nonpolar compounds mix with fats. So the major nonpolar thing that we think about a lot of times is the membrane, which is, of course, primarily made of phospholipids, and so it is a nonpolar thing. Some other chemistry that could show up, remember, these are the main 2 types of reaction that we see in a living system. Hydrolysis, and just go back to your word roots from the first month of school, right? Hydrolysis, using water to split something or break something down, versus dehydration synthesis. So again, taking away water to dehydrate will allow something to come together or be put together or synthesized. Something else that I want to bring up, mainly just in light of metabolism, is oxidation and reduction, right? Remember oil rig, like this pretty little oil rig here out in the ocean. Oxidation is loss of electrons, while reduction is gain of electrons. So if a molecule loses electrons, it is being oxidized. This typically involves breaking it down, right? If something is reduced, that means it's picking up new electrons, and that's often involved with sort of building up things. So, for instance, when we talk about breaking down the sugar in metabolism, we are oxidizing it, right? We're taking away its electrons. And then here's an oil rig that is releasing energy. So also don't forget about these kind of generic terms, but an exergonic reaction would give off energy into its surroundings, while endergonic reactions will take in energy from their surroundings. All right, and last thing about chemistry, sort of 2 things. One is thermodynamics. This is really something that we spent more time talking about when we got to metabolism, because it just deals with energy. But technically a part of biochemistry or thermochemistry. But remember the laws of thermodynamics, right? Energy is not created or destroyed. It just changes forms in our systems. So again, if we think about metabolism, we're going to take a sugar, that chemical energy, we're going to use it to make cellular energy, but we're also going to lose some of that energy as heat. It's just a changing of forms. And all that takes place because energy transfer is very imperfect, which is sort of the second law of thermodynamics. And that second law, if you recall, tells us that we are constantly becoming disordered, right? And so what your body is going to do is use energy to decrease that disorder, or what we call entropy, and bring about order. So we want to have organized, ordered systems, whether it's a cell or an organ or a body or whatever. And so we're going to have that constant input of energy to decrease disorder. And what we talked about briefly in class, classic Berenstain Bears book here, is, our room or our house just sort of inherently becomes messy unless we invest energy in focusing on keeping it clean. And that's what your body and your cells are doing, is they're investing energy constantly to keep your systems ordered and fully functional. And then we got water. Again, I'd kind of be shocked if that really shows up on this year's test. But water is kind of important, right? It's the major solvent on our planet because it is polar. It provides surface tension. It provides capillary action, which allows fluids to move through tubes. Again, I don't know that water will show up on there. Fun, quick quiz. This is a Marvel character. Does anybody know who it is? He's rumored to be showing up sort of in the next phase of Marvel Cinematic Universe movies. So this is a rendering. They don't have an actual actor tabbed yet to play this, but they've dropped little hints in Iron Man and particularly in Black Panther that this guy could show up. His name is Namor the Submariner. And so the Submariner is sort of Marvel's version of Aquaman, who comes from DC, right? But Submariner, really old comic book character that might be showing up soon in the Marvel movies. Now, topic infinity I put here. I'm kind of being a smartass, but is feedback regulation. Remember, feedback regulation is, again, potentially going to be on the test, but it's the biggest concept of the way we would control almost any system, right, or any pathway in our bodies or in any kind of living scenario we're talking about. So, again, any pathway we've talked about this year, like I put up in this box, probably involves feedback control, whether it was cell signaling or it was metabolism or more recently in the endocrine system with those specific examples. We are going to use, feedback in our systems, and this allows us to regulate processes. It allows us to maintain homeostasis within those processes, right? And in feedback regulation or feedback control, the product, or maybe it's the response in the cell signaling cascade, feeds back and affects the earlier stages of the pathway. So negative feedback, much more common, right? Sometimes also referred to as feedback inhibition. This is where your resulting product or your resulting response goes back and turns off the pathway. Also possible is positive feedback, however. We just saw this in the endocrine notes, which is where maybe sometimes the result actually amplifies the process or keeps the process going, keeps the pathway running. So again, negative feedback much more common because negative feedback tends to kind of bounce us back and forth, but keep us right around that homeostatic level where things started. Now, topic 6, and I know this is getting really long. We only have a couple more here, though, to take care of the first quarter. We did a ton. Did you realize how much we did in the first quarter? Topic 6, prokaryotes versus eukaryotes. This is just basic cell structure. This certainly could come into play. This is something that I almost feel like the AP people would think is too easy if they only have 2 questions to throw on there, but maybe not. But remember, all cells are membrane enclosed, so all cells have a cell membrane with cytosol inside there. All cells also have genetic material, typically in the form of DNA, and all cells have ribosomes. So if you're asked about something that is common to all cells, think membrane, genetic material, and ribosomes. Then when we look at prokaryote versus eukaryote, put this table down here. This is, I believe, appeared in our notes previously, but just a good comparison. Biggest thing that is different, right, is a prokaryotic cell does not have a nucleus, whereas a eukaryotic cell does have a nucleus. And a eukaryotic cell also has a bunch of particularly complex compartmentalizations, right? Organelles that we see there. And so tiny organelles inside the cell. And you do want to know sort of the gist of each of these. I wouldn't get real caught up in some in-depth description of what they do, but you certainly want to have some familiarity with it. So like mitochondria, try to go beyond powerhouse of the cell, right? But this is the site of respiration, so energy processing. Lysosomes, breaking stuff down inside the cell. The Golgi as well as the ER, both there for processing of products that are created or moved within the cell. Ribosomes make protein, right? But almost just as valuable, remember this is the site of translation if we're talking genetics. Cell wall, extra protective barrier found in plants and fungi, but not found in animal cells. And chloroplast, site of photosynthesis, but found only in plants and algae, and again, not in animal cells. And then I put this little box up here too. Don't forget about endosymbiotic theory. I could see this, not being the crux of a question, but maybe just being a part C or something. Remember, endosymbiosis suggests that a eukaryote came to be because one prokaryote kind of swallowed or engulfed another, right? And the larger prokaryote became the cell, and the smaller prokaryote became a mitochondrion, or became the nucleus, or became a chloroplast. And again, there's evidence to support this because chloroplasts and mitochondria have their own DNA. They have a double membrane system. They can reproduce separate of the whole cell, things like that. Now, topic 7, membrane exchange, moving things across the membrane, right? Remember that the membrane is selectively permeable, meaning that it allows some things through, but not others. And so we've got diffusion, right? Diffusion being the movement of molecules from high concentration to low concentration, or in other words, molecules down their concentration gradient. This can be simple diffusion, which would mean directly across the membrane, right? Or it can be facilitated diffusion, which would mean that we need to have some sort of channel or transporter or something to allow things through the membrane. Remember that the rate of diffusion is affected by lots of stuff. Charge is bad, right? That will, a charged molecule will decrease the rate of movement across the membrane. Size, right? Big is bad, right? Polar is bad. Concentration gradient, the steeper the better. So the bigger the difference is between concentrations, the more diffusion you're going to see happen. Surface area, more surface area is a good thing. It's more area of the cell that the molecules can move through. Temperature also higher is better. Again, we think about an increased molecular movement when we increase temperature. And then also pressure, right? Pressure can be positive or negative, whether it's a push or a pull in moving molecules across. And we also have this little graph I copied over up here. Remember that simple diffusion or non-mediated diffusion here in this picture can just kind of increase exponentially, whereas facilitated diffusion will hit a limitation, right? If you have to use a channel, all those channels can be full or occupied, and so this one does have a tendency to level off. Then don't forget we have a couple other types of transport as well. Remember, active transport requires an input of energy. This is more what we would think of as pumps, because they're going to push molecules against their gradient, and that's what makes this different than basic diffusion, is that we're going to use energy to push things up their concentration gradient. Bulk transport, brought this up recently in the immune system, but bulk transport means bringing in or giving off a large amount of molecules at a time. So we can do endocytosis or phagocytosis, would be engulfing a large thing like a whole other cell, versus exocytosis, right, would mean out, right, so, releasing something from the cell. And then really important but basic concept, right, is just don't forget about equilibrium being dynamic. Molecules will never stop moving. It's an impossibility. So molecules will constantly move, and what we look for is we get to the point where things are kind of equally moving back and forth across the membrane. And that is what true equilibrium is. Now, you knew it was coming. While we're on the topic of membrane exchange, we have to talk a little bit about osmosis, and that means that we're eventually going to walk down this horrible dark path to water potential here in a moment. But remember, osmosis is the diffusion of water. Water molecules also move down their gradient, but remember that we don't typically talk about, the concentration of water molecules. Rather, we talk about the concentration of solute or stuff dissolved in the water. So water will move toward the higher concentration of solutes, and so we talked about tonicities. Now, big thing to remember, right? Cells are not tonic, so you never want to say that the cell, you never want to say that the cell became hypotonic or something like that. That just doesn't happen. It's the solution, right, around the cell that has a tonicity. And so remember, isotonic, iso meaning same, means that the cell and its surroundings have the same concentration of solutes. Hypotonic, hypo meaning under or less than, right? This means that the solution out here has less solutes than what we see inside the cell. And hypertonic, hyper meaning more, right, means that the solution out here has more solutes than what is inside the cell. And then maybe what's most important is, can you predict what a cell will do in one of these scenarios? So let's look at another picture. So when we talk about animal cells, right, animal cells typically prefer to be in an isotonic solution where you have this equal flow of water in and out of the cell. If we drop an animal cell into a hypotonic solution, think hippo, right? Water will move toward the solutes, so it will move into the cell. The cell will swell up and potentially burst. Versus if we put an animal cell into a hypertonic solution, water will rush out of the cell to get to that higher concentration of solutes on the outside, and the cell will shrivel up, and we called this crenation in the example of red blood cells, which is also just as bad as bursting, basically. But then don't forget that plant cells are different, right? Plant cells with their cell wall run by sort of a different set of rules. For a plant cell, hypotonic is best, a scenario that they are well watered and their water will rush into those cells and make those cells turgid or fill those cells up. Isotonic solutions aren't terrible for plants, but they're also less than ideal, right? So again, equal flow of water in and out is okay. Hard to see the green here, sorry. Is okay, but does tend to leave us with cells that are somewhat flaccid. And then worst case scenario for a plant is a hypertonic solution, so a really salty environment or something like that. You're going to see water leaving the cells. The cells will become plasmolized, which again, remember, means that the membrane here actually pulls away from the wall, and that's not a good scenario, right? The cell probably can never reattach that, and so the cell will most likely die. One just thing, make sure you keep in mind is that plant cells will not burst. So you don't want to suggest that either. It would take a ridiculous amount of pressure to burst a plant cell because of its cell wall. Here we are, water potential. So I put my man T'Challa down here, who's basically saying, "Bring it on!" Right? Bring on those tough questions about water potential. So remember, water potential. Again, I'll be honest. I think I'd probably be surprised if water potential shows up on this test with only a couple of questions on it. But just in case, right? Water potential is another way to predict water movement. It involves solute potential and pressure potential. Remember that solute potential is always negative. Adding a solute here will, so to speak, remove the water molecules from the solution. So it gives us a negative potential. Pressure can be positive or negative, whether it's a push or a pull. But most importantly, remember those key pieces, right? Water moves from high potential to low potential. This is, a very basic statement, but it's very important to know. Now, also I put on here, low potential usually means negative, right? And again, water moves to where there's more solute concentration. So it makes sense, right? If solutes make things negative in terms of water potential, then it makes sense that water will move to where there's more negative. Pure water is going to have a potential of 0. Having solutes, as we already said, is going to make that water potential lower. And then pressure can be pushing or pulling. So if we use this little picture we had previously in our notes, the pure water outside of this cell, can't really tell there's anything in here, but let's say this thing's full of water. In this water out here, the water potential is 0, right? There's no pressure because it's an open container, and there's no solutes. But the potato cell here in the picture also is not under any pressure initially, but it does have a solute concentration because it's got some carbohydrates and stuff in it of -3, which means that we have a water potential of -3. So in this scenario, if the potential is 0 outside and it's -3 inside, water will move to the lower potential. So water would move inside the cell, right, and hopefully make this potato cell a little bit more turgid. Now, don't forget that a couple things, right? One, don't forget that water will sort of cease net movement. Again, it never stops moving, but it'll cease net movement. So we'll say it stops when the potential inside equals the potential outside. But keep in mind, too, which is what we've got here in this example, that there's sort of more than one way to establish water potential. Like I put on here, solutes might give you a solute potential of -3. But eventually, that's going to draw water into the potato cell and build up a turgor pressure. Right? And maybe that pressure pushing against the cell wall becomes +3. So now check out back to my picture, right? If this turgor pressure builds up to get to +3, now we've got +3 plus -3 is just going to give us 0. And if we've now got 0 in the potato cell and 0 outside the potato cell, we have no more net movement. Right? And then the last thing is just in terms of sort of its practical application, maybe water potential we mentioned more recently is involved in moving water up a plant, so up through its vessels, and that involves transpiration. So transpiration or evaporation of water from plant leaves gives a pulling force. It sets up this kind of gradient of water potential stretching from those leaves all the way down to the roots. And the most negative potential is up there at that leaf to air interface. And so, we're going to see this plant, and we are going to see the fluids or the water being pulled up the plant because of transpiration at those leaves. All right, I promise we are getting close to the end of this massive Q1 review document. But we do have one more big topic that we hit not long before the end of the first quarter, and that's cell signaling. Also one of the obscure but one of my favorite characters from the Marvel Cinematic Universe here, Korg, who first appears in Thor Ragnarok and then makes some little little appearances later as well. But Korg is great, right? As you can see, I'm made of rocks, but don't let that intimidate you unless you're made of scissors. Right? And then he follows that with just a little rock, paper, scissors joke for you. But cell signaling, right? Cell signaling, I think, incredibly important. I'm guessing you're kind of picking up on that because signaling has been a part of several of our practice free response questions so far. And again, it's not that the entire question has been devoted to cell signaling, but it shows up just in one little part of the question. It feels like almost every time they write one. And so a really important thing. I would not stress too much on heavy detail, right? As was the case with almost everything we did this year, I pushed you to a higher level of detail than was absolutely necessary. And the goal there is that you remember or retain enough of the stuff in the end that it comes back to you and that you don't necessarily retain all those details. And so now that is also still true, right? I would definitely know the 3 general stages here of cell signaling. But beyond that, I mean, we had a number of slides about each one, and we can kind of compact this into 1 or 2 slides now, right? Reception, right? You have to have a receptor, whether that's on the cell surface or it's inside the cell, and you have to have a ligand, any kind of molecule that binds to a receptor. That's how the signal gets received by a cell. And the most common type of receptor was the G protein coupled receptors that are lodged in the membrane of the cell. We did mention receptor tyrosine kinases, or RTKs, although they're not nearly as common. And we also mentioned, especially recently with the nervous system, ligand-gated ion channels being another thing that's a type of receptor that can bind with a ligand. But after we have reception, we go to transduction. Remember, transduction involves a phosphorylation cascade. So one molecule basically adds a phosphate to the next molecule, which adds a phosphate to the next molecule, and so on and so forth. This is the result, and we'll see a picture of this in just a moment, of kinase enzymes, right, that can do this phosphorylating or this phosphorylation cascade. We also mentioned second messengers, probably not as critical of a piece of knowledge right now at this point, but cyclic AMP is an example of a second messenger. Calcium is an example of a second messenger, which is just a member of the transduction process that's not a kinase, right, and not necessarily a phosphorylating event. And then you got response, right? And responses can be nuclear or cytoplasmic, right, or sometimes we say cellular. Remember, nuclear response is where we're going to go in and we're going to regulate transcription within the nucleus, so whether or not you express genes. Cytoplasmic or cellular responses are where we're just going to do something else in the cell, right? Maybe we open some channels to change or affect diffusion. Maybe we mess with the cytoskeleton a little bit. Maybe we activate a particular enzyme. Maybe we trigger or slow down cell division. Who knows? But if it's out somewhere other than the nucleus, we would call that a cytoplasmic or a cellular response. There's a great slide just about kinases and phosphatases. Right? Remember that a kinase is a phosphorylating enzyme. So it is going to, this is really hard to see in this picture, but it's going to, add a phosphate to some kind of protein, and it's going to get that phosphate from ATP, right? So it's going to take this phosphate from ATP, resulting in ADP, and add that phosphate to our protein or phosphorylate it. But then it's also just as important, actually let me back up for a second. Remember, this is typically an activation of a signal. And it's also just as important to be able to deactivate. So deactivation of a signal. And that's what a phosphatase enzyme can do. So again, it's really hard to see these in the yellow. But if we take a phosphate away from, right, a protein that's performed by a phosphatase, that allows us actually to create ATP. So we give that phosphate over there to that guy. And again, this would turn or shut off a pathway or a signal or a system. Last thing about cell signaling, one of my favorite topics of anything is apoptosis. Apoptosis is, again, sort of programmed or intentional cell death. It is, sounds bad, but it's also extremely important for development and growth, right? As we develop and become what we are with the structures that we do have. It's also very protective, right? If a cell becomes bad or if a cell goes rogue, apoptosis hopefully gets rid of that cell. So, for instance, like in a cancerous situation, if a cell takes on some cancerous changes, we hope that that cell will remove itself from the population. Here's what that looked like, right? Remember, an apoptotic cell will bleb, which means it sort of just breaks itself in these little vesicles that can then be engulfed by an immune system cell here, which would just get rid of it. This involves signal transduction in a really complex pathway that we don't have to know a lot of detail about, but the enzymes in here are called caspases. Caspases will break down parts of the cell. Again, caspases are a specific example of a protease. So protease being, any enzyme that breaks down protein, hence the name. So you got these proteases called caspases, which destroy the cell. And there's a couple of reasons why you might see apoptosis. Might be nuclear response, so a nucleus notices that there's some kind of mutation or some kind of DNA damage. Also might be that the endoplasmic reticulum notices some misfolded proteins. In either case, we set off this signal transduction cascade that causes the cell to kill itself. And remember, a couple real-world applications. In a cancerous situation, which is too much cell growth, apoptosis is probably lacking, right? We should be killing off these cells, and we're not. Also, we think that apoptosis might be overactive in some degenerative diseases. So we talked a little bit about like Alzheimer's and Parkinson's in the brain, but if there's parts of the body that are breaking down, it's possible that there's too much apoptosis there. And then I absolutely love this. This is probably my favorite Guardians of the Galaxy picture. Mainly just I like Rocket's reaction, and I really like Drax and Groot over here too. All right, now the last couple things here. I promise this stuff's easy here. Topic 9, mitosis versus meiosis. So remember, this is cell cycle, right? But the most important thing about the cell cycle is that you want to make sure you know that difference between mitosis and meiosis. Remember, if we're just talking about the cell cycle, remember that the majority of that cycle is interphase. So if we were to draw the cell cycle over here, remember that, we might have this tiny little wedge, which is, representative of our M phase or our mitotic phase. And then the rest of this big thing is all going to be interphase. And remember, interphase has really kind of 3 stages to it. We got G1, we got S, and we got G2. And remember, this is controlled by checkpoints. And so basically, we've got these checkpoints where, a cell, won't move into the next stage without clearing the checkpoint. And remember that involves cyclins and CDKs. So cyclins were constantly fluctuating, right? And CDKs were at a consistent, or these are kinases, right? But the CDKs are only active when there's enough cyclin. And so the cell will raise or lower cyclin levels in response to what's going on. And if the cyclin levels are high enough, then it continues through the cycle and eventually marches its way into cell division. Now, again, if we're going to compare mitosis with meiosis, remember, mitosis involves prophase, metaphase, anaphase, and telophase, followed by a cytokinesis step where the cell splits. Remember that meiosis really follows the same kind of pattern, but it does so twice. It's a double division. And then remember these differences in the results, right? Mitosis is going to give us 2 daughters, while meiosis gives us 4. Those 2 mitotic daughter cells are diploid, just like the parent cell we started with, whereas meiotic resulting cells are haploid, or only have half the DNA. And so these are our gametes, right, our sex cells that we use in reproduction. And then remember that mitotic daughters are identical to each other and the cell we started with, while meiotic daughter cells are genetically unique. And so here is this big, massive chart from your textbook, but does a fantastic job. I mean, you could just study this chart and you would know mitosis and meiosis of showing you the stages and things. Again, I wouldn't get real caught up. I don't think you're going to have to draw a picture of this or anything of the sort, but you definitely want to be able to compare the two. And then there's a sweet picture of all the tough ladies about to lay the smackdown in Avengers Endgame. And then the last slide here, right before the end of our first quarter together, we looked at the difference between mitosis and meiosis. And so, again, meiosis has 3 particular events that are unique to it. That involve synapses, or the joining of the homologous chromosomes, and crossing over, or technically referred to as genetic recombination. All right, meiosis 1, we saw homologous pairs at the metaphase plate versus just a single file set of chromosomes. And during anaphase 1, we saw separation of those homologs versus any kind of separation of sister chromatids like we would see in mitosis and we would see in meiosis 2. Also, then remember we had a small PowerPoint that was sort of all about genetic variation and how important genetic variation is. All right, remember that genetic variation is really what allows us to evolve. Right? If we don't have those differences in our traits or in our characteristics, then there can be no such thing as natural selection. Right? But if there are different organisms out there, and again, just kind of sticking to a very basic understanding of evolution and natural selection here, but if there are different types of organisms out there, then it makes sense that some of them will be better at surviving than others, and it maintains our population much longer, right? So where do we get genetic variation? Remember that chromosomes line up independently. So how they line up is never really predetermined, who's on the right, who's on the left. We follow that up with crossing over, or genetic recombination, which is going to jumble up your genes and give you offspring, or give you gametes here that are totally unique. And then you also have random fertilization, which technically the textbook talks about this in the sense of we never know which sperm will fertilize which egg, but I would even go so far as to suggest we don't know who that sperm comes from either, which is a whole nother almost innumerable number of potential things there, potential outcomes. So it gives us a ridiculous amount of variation when organisms sexually reproduce. And again, this is super critical to populations not going extinct on our planet. And so we finished up with a sweet Avengers picture of all the different people, including Captain America wielding his broken shield and Mjolnir, the hammer of Thor. So I know that was a ton of stuff, but hey, at least it's all in one place, and hopefully it helps kind of refresh things. And now with the month you have until the test shows up, you can whittle through a lot of this stuff and start looking at these notes. So thanks for hanging with me.